Hello guys and welcome to the Biology Seeker YouTube channel. Today's topic for the video is COVID-19 and bioinformatics. Okay, so the term coronavirus came from the Greek word which is corona, and that means a crown. So as you shown in the as you shown in this the as you shown in the figure, the coronavirus has different proteins like spike protein, envelope protein, membrane protein, nucleocapsid protein, and tricks. So we can contribute. So the coronavirus first attached with the AC receptor, which is present on the human ciliated epithelial cells, which are mostly present in the lungs. So after the attachment with the receptor, the coronavirus get entry in the cell. Okay. After the getting entry in the cells, coronavirus hijack the replicatory machinery of the host, and after it start replicating itself. Okay, so in the replication process, first the coronavirus genome get translated to become to first it is translated because the coronavirus has some outer, as we seen, the coronavirus has some outer proteins, so it has to make some outer proteins again for new generation. So first it is translated. First, the coronavirus genome is translation of the coronavirus genome is occur, and it is produced two main proteins, which is known as pp1a and pp1b, and the proteolysis of these proteins occur. After proteolysis of these proteins, they can act as a replicase enzyme. So after this, the transcription of the gene occurs, and further the translation occurs as we know of the central dogma of life. So after the translation, the proteins which is known as spike protein, envelope protein, and membrane protein are translated to the Golgi, and after the assembly and packaging, the virus get released from the cell. Okay, as we shown in here. So now bioinformatics and coronavirus. How can we use bioinformatics for coronavirus prediction and other things? So, as shown in this figure, there is a nucleotide chain, nucleotide sequence of 30,000 genes, and around in 22,000 to 26,000 there is a region for spike protein sequences. Okay, so after it we can we can get the amino acid sequences of this nucleus this nucleotide sequences, as is shown in the S1 unit in the figure A. There is a mutation in contact residues of the SARS-CoV-2 spike protein. The spike protein of the SARS-CoV-2, which is red bar at the top, was aligned against the most closely related SARS-CoV like coronavirus and the SARS-CoV itself. Key residues in the spike protein that make contact to the AC2 receptor are marked with blue boxes in both coronavirus 2 as well as and its related virus, including the SARS virus also. So is the including the SARS coronavirus also, which is, so as we seen in this figure, the different sequence alignment occur, and there is some amino acid changes in the sequence of the novel coronavirus, as we seen here. Okay, okay. And in the S2 subunit, there is uh, acquisition of polybasic cleavage site and olink glycan residues. Okay, both the polybasic cleavage site and the three adjacent predicted olink glycans are unique to SARS coronavirus and were not previously seen in lineage of beta coronavirus. Okay, so as we know that naturally the coronavirus get mutated, and in its polybasic cleavage sites here, actually here in this region, this region, so it is not clear, but in this region the polybasic cleavage site of the other coronavirus are RRAR, but the addiction of the lysine residues in the novel SARS coronavirus, which is here, is a lysine molecule. Okay, so due to the lysine molecule addiction, the polybasic cleavage site is changed. Okay, and the functional consequences of the polybasic cleavage site in the SARS-CoV-2 is unknown. Okay, and all the function of, and there is also Olink glycans, and the function of the glycans is unclear, but they could create a mutation like domain that seals epitopes or key residues on the SARS-CoV-2 spike protein. And with this mechanism, several viruses utilize mutation like domain as a glycan shields to involved in the immune evasion mechanisms, which we talk about it later in video. So, and the other is the metagenomic analysis. Okay, so in this metagenomic analysis we take the sample from the different peoples, around 1578. Okay, and after the analysis we came to what we do in experiment. First we take the sample of the all patients which has the symptoms like coronavirus, like the flu and flu or coronavirus. So firstly it is done in prior and in January when the outbreak came, when the outbreak in the Wuhan. Okay, so we take around 1578 different samples from the nasal swab only. After it after the analysis of these samples, we do the PCR test. Okay, so with the PCR test we are able to we are checking for the presence of the SARS-CoV-2 genome, and from 1578 samples, the 1378 samples are seems positive for the coronavirus. So basically we came to know, yes, this is a novel coronavirus, and it is get outbreak. Basically it is show that the more than 90% of the samples came positive for the SARS-CoV virus genome. And in B, we have seen the different open trading frames for genome for coronavirus genome and yes. So as we seen in the figure C, we found that a short region of RNA dependent RNA polymerase from bad corona, which was previously detected, showed high sequence identity to the novel coronavirus 19. We carried out a full length sequencing on this RNA sample. Simplot analysis showed that novel coronavirus was highly similar throughout the genome of the bat RAG13. Okay, with overall genome sequence identity of 96.2% using the aligned genome sequences of 2019 coronavirus, RECTG3, and all of this, the file and all of this, there is no evidence of for recombination events was detected in the genome of 2019 novel coronavirus. Okay, phylogenetic analysis of the full length genome showed that which is from the Wuhan is basically show the high similarity with the bad cove rect TG13. So it is similar with it. Yes, so it is similar with it. Okay, and then next slide. So some companies or research groups are actively finding drug or make a vaccine for this. So first is ABV company. This company used the ritonavir drug, which is a HIV protease inhibitor drug. This drugs uses mainly this drug affected the proteolysis mechanism of virus. Okay, so we can use this old drug also in the coronavirus, but not But the prediction or precision medicine, for as we can use this drug or where this drug as a novel agent is not now known, is not much known, but we can use this drug. Second is athletics pharma company. This company used two different drug in combination, and Danoprevir and Ritonavir. Both are HIV. Both are HIV. The Ritonavir, as I told you again, as I told you in the as I told you ago, the ritonavir is the HIV protease inhibitor, and the danoprevir. This is a drug which is the same mechanism like ritonavir, but it is useful in the HCV infection. So this pharma company do trials for the combination of those both drugs to prevent the COVID-19 disease. And the third one is Bernoulli group. This group use the nanobody based treatment, or we can use nanoparticle, or the nano based treatment. And another treatment is for another drug is the nanoparticle drug. With the nanoparticle delivery, we are fooling the virus that the nanoparticle has the similarity with the ACE2 receptor. Sorry, not protein, ACE2 receptor. So the coronavirus can bind with the receptor, and not man the mouth infection in the cell is not occur. But it is also in clinical trial, so we hope for the best. And thank you for watching the video. And we also have an announcement for you. We are introducing a new course for introduction to next generation sequencing, which is coming soon. So the students who are interested in, please contact at info.biologyseeker@gmail.com. Okay.